Good afternoon, everyone. My name is David Eaton. I welcome you to the NTP Board of Scientific Counselors meeting for today, December 15th, 2022. The first order of business after calling us to order is for the board members to introduce themselves, and I will start in a moment. I will note that unfortunately a number of our board members today are not able to join us for a variety of reasons. Dr. Wei-Shui Chu, Kathleen Gray, Matthew Martin, Susan Tilton, and Pam Lyon are not able to join us. My name is David Eaton. I am the chair of the BSC, and I am an emeritus professor of environmental and occupational health sciences at the University of Washington and adjunct professor of pharmacology and toxicology at the University of Arizona. We will go down the list here, and I would ask that each member just briefly introduce themselves where they're at, and maybe even a word about their area of expertise. I should mention that my background is in toxicology, and particularly in the area of chemical metabolism and chemical carcinogenesis. Dr. Berube.
I am a professor of science and technology communication at North Carolina State University. And over the last 2 years, I've written a book on pandemic communication, another on pandemic resilience and messaging we should have learned about Zika to deal with the COVID currents.
Thank you. And congratulations on your important book as well. Eric Bloom, Dr. Bloom.
Good afternoon. So Eric Blum, I'm Vice President of Development Biological Sciences at AbbVie, located in the Chicago area. So development biological sciences covers area like pathology, toxicology, drug metabolism, bioanalysis, and lab animal resources.
Thank you, Eric. And let's see, Wei-Shui is not here, so Dr. Felter. Susan?
Hi, I'm Susan Felter. I am a research fellow in Procter & Gamble's Corporate Human Safety Organization, and I specialize primarily in chemical carcinogenesis and risk assessment.
Thank you. Dr. Michaels.
Hello, David Michaels. I'm a professor at the George Washington University School of Public Health. I'm an epidemiologist. My particular interest is in occupational health. I ran the Occupational Safety and Health Administration for 7 years earlier in the last decade, excuse me. And I've written a couple of books about scientific integrity and ensuring the veracity and integrity of the science that we use for public health and environmental regulation.
All right, thank you, David. So Dr. Payne Sturgis, Devon?
Yes, hello everyone. Good afternoon, everyone. I'm Devon Payne Sturgis. I'm an associate professor of environmental health sciences at the University of Maryland School of Public Health. I'm really inspired by all these book authors among us. That's terrific. My areas of interest include children's environmental health, cumulative risks and cumulative impacts, and policy reform.
Great. Thank you, Devon. Mark Russi, Dr. Russi.
Hi, Mark Russi. I'm a professor of medicine and epidemiology at Yale University. My area of expertise is occupational and environmental medicine, particularly that which is applicable to healthcare workers.
Thank you. Dr. Ann Ryan. Ann?
Hi, I'm Ann Ryan. I trained as a veterinary anatomic pathologist, and prior to my retirement, I was head of an investigative toxicology department at a pharmaceutical company.
Great, thank you, Ann. And last but not least, Veena Singla, Dr. Singla.
Hi, good morning, good afternoon, everybody. Veena Singla with the Natural Resources Defense Council and the People and Communities Program. And my work seeks to address health disparities to harmful environmental exposures and bring the most current science and data to inform better decisions. Thank you.
Thank you, Veena. And now I would like some of the NTP leadership that is here to introduce themselves. And I believe, is Dr. Wojcik with us?
He's not, Dave.
That's what I thought. Okay, so Dr. Berridge, Brian, go ahead.
Yep, thanks, Dave. So hi, everybody. Good morning, good afternoon. I'm Brian Berridge. I am the Scientific Director for the Division of Translational Toxicology here at NIEHS, and also the nominal Associate Director of the National Toxicology Program.
Thank you. And Dr. Wolf, Mary?
Good afternoon. I'm Mary Wolf. I'm the Director of the Office of Policy Review and Outreach and the Acting Division Director for Outreach and Communication. Thank you.
Thank you. And Michelle, Dr. Huth.
Hi, everyone. I'm Michelle Huth. I'm the Acting Deputy Director for Program Operations.
Thank you. Dr. Brownlow.
Hi, everybody. I am Melanie Brownlow. I'm a health scientist in the Office of Policy Review and Outreach in the Division of Translational Toxicology. And I am also the designated federal official for this meeting. Welcome, everybody.
Thank you, Melanie. We have two of our liaisons with us from the FDA, Dr. Goncalo Gamboa da Costa. Dr. da Costa.
Good morning or good afternoon, everyone. Yeah, my name is Gonçalo Gamboa da Costa, and I'm the FDA Liaison Officer to the National Toxicology Program.
Thank you. And Dr. Christina Lawson from NIOSH. Christina.
Good afternoon, everybody. I'm Tina Lawson from NIOSH and liaison to the NTP. Thank you.
Thank you. So it's my pleasure now, I believe, to turn this over to Dr. Berridge for some comments. Brian?
It is. Thanks, Dave. So again, welcome everybody. And on behalf of Dr. Wojcik, we appreciate your attendance and your participation and your support through this BSE meeting. This is a meeting that will be very much like the one that we had back in April, where we will be reviewing some contract concepts. That's an important function of this Board of Scientific Counselors, so I appreciate you all's dedication. I'd like to also take the opportunity to recognize some of the members of our esteemed BSE who will be completing their terms and rotating off. As you all have come to recognize, this is an incredibly well compensated service that you all provide. And I say that tongue in cheek because it's not very well compensated, but it is an important function and we very much appreciate your service. Typical term for a BSE member is about 4 years, and several of you have paid your debt to society and served us by supporting us through that 4 years. And so I'd like to just sort of recognize each of you. So first off, Dave Eaton, who has been our fearless leader for a number of years as the chair of the BSE, has completed his term. I have to say that Dr. Eaton will not be getting off that easy because he will continue in a role in chairing a very important working group that is currently active. So he'll be with us for a bit longer, but I'm sure Dave will be looking forward to his retirement from the BSE. So Dave, thank you for your service and your continued service.
Thank you,
Dr. David Berube. So Dr. Berube has brought some unique perspectives to this group over the last few years, very much aligned to his area of specialty, which I think is increasingly in importance to us in science communications. So it's been a valuable member to the group, and I really appreciate all of your input, Dr. Berube. Dr. Susan Felter. So thank you very much, Susan, for your contributions, bringing an important industrial perspective, but always also very critical and insightful comments to the group, and very much appreciate your service and support of the BSE. Dr. David Michaels, who has spanned both government service and academic service, and over the years has provided great insights for us too. So appreciate your service, David. Dr. Ann Ryan. So Dr. Ryan is a colleague of mine who I've known for a number of years, and very much appreciate your participation in this BSE and providing your insights. So thank you very much for that, Ann.
Sure.
And then we have two members who are also rotating off, who are not in attendance. Dr. Wei Shui Chu from Texas A&M University, very much appreciate his service, and Dr. Susan Tilton from Oregon State University, who's also not in attendance. So in addition to the incredible compensation we provide, we do also have a certificate to recognize your service. And so that will be put in the mail I'm sure displayed prominently in all of your living rooms. So thank you again for your incredible service. I can't begin to tell you how important it is for you all to get your perspectives. And honestly, over the last few years, we've put you through a ringer because we've introduced you to our entire portfolio, particularly in 2021, where we infringed on your good nature to review concepts for our entire portfolio. And so that was very much appreciated. And in advance, I appreciate your service today in providing some support in these review of our contract concepts. In addition to that, and unfortunately, Dr. Line is not here, but was also wanted to recognize her significant achievements. She was recently awarded the American Association of Veterinary Medical Colleges Excellence in Research Award, which apparently she will receive in a ceremony at their meeting in 2023. So it's all of you are incredibly accomplished folks and well known in your field, and it gives significant credibility to our organization to have you advising us and supporting us. So again, very much appreciate that. Dave, back to you.
Thank you, Brian. And I would just take this moment to also thank all of the outgoing board members and actually the whole board that I've had the pleasure of working with for the last several years. And it has been some challenging times with COVID and a lot of virtual meetings that in the end, I think have gone very well. And Brian, I'd like to thank you for your leadership of the NTP in these last few years that I've been engaged here. So with that, we will move on to the agenda. I will ask Dr. Melanie Brownlow for a conflict of interest statement and other housekeeping items. Dr. Brownlow.
I think you're on mute.
Thank you. Sorry about that. Thank you, Dr. Eaton, and thank you for the reminder. The members of the NTP Board of Scientific Counselors serve as individual scientists and not as representative of any organization. Each member is to exercise judgment prior to or during the meeting as to whether a potential conflict of interest might exist relative to the topic under discussion due to his or her occupational affiliation, professional activity, or financial interests, or those of their spouse, minor child, partner, or organization. Board members are asked to bring to the attention of the committee and of the federal official any potential conflict of interest that might pertain to the topic being considered at the meeting. Should there be a potential conflict of interest, it will be handled in accordance with departmental policies and regulatory requirements. Thank you very much. And next, I'm going to read, just going to go over a few housekeeping items. So this is a public meeting that's being broadcast to a remote audience. So when speaking, please remember to introduce yourself for the listening audience. And additionally, since this is a public meeting, we ask everybody to reserve the use of the Zoom chat box to technical issues only. All discussions in the meeting will take place in the public forum and are going to be captured in the meeting minutes. So we also ask the presenters and speakers to mute yourselves and turn off your video unless you are speaking as part of Zoom etiquette. So lastly, slides will be made available on the website as soon as they meet government requirements for accessibility. In the same way, minutes will be posted to the website once they have been reviewed for accuracy by all the BSC members and approved by the chair, and then made available on the website as soon as they meet the government requirements for accessibility. So with that, I'll turn it back over to our chair. Thank you.
Thank you, Dr. Brownlow. So it's my duty to acknowledge that there were no public comments received on any of the agenda topics and no written public comments and no oral comments were requested. With that, I would note this is an important meeting because there are three contract concepts which the Board of Scientific Counselors is required to review and approve all contracts through the NTP. And we have three of those today, one on chemistry support services, another on toxicological support services, and the third on pathology support services. And obviously, each and every one of these areas is integral to the function of the NTP and their research programs. With that, I think we will begin with the presentation from Ms. Julie Bardot on the chemistry support services for the NIEHS. So with that, I will turn this over to Dr. Stout, I gather.
Yes, good morning or good afternoon, as the case may be. I'm going to present some introductory material that hopefully you'll find useful and informative for the discussions of the specific contract concepts. Next slide, please. So in terms of contract concepts, what is to be reviewed by the BSC? So we're talking about research and development contracts that are either new or are recompetitions with changes in the statements of work, or that have been ongoing for 5 or more years since the last review. And those last two bullets in bold are the situations that cover the contract concepts for today. This review is required prior to issuance of a request for proposal. And I think it's important to note that a single contract concept may encompass multiple statements of work, solicitations, and or ultimate contracts. And the resulting contracts will be with the NIEHS. However, these may also support NTP projects. Next slide, please. So I wanted to talk a little bit about the allocation of the Division of Translational Toxicology, Toxicology's funding. So we have two major types of funding, or two types of funding. Intramural or IR funding, which covers things like salaries and fringe benefits, operating expenses, and travel. Within that, we have two intramural laboratory functions that do a lot of in-house research. The major part of our budget, which is about 3.5 times larger, is our research and development budget, which funds contracts and interagency agreements. And there's not a lot of sort of crosstalk allowed between these two types of funding. So our these essentially fund separate functions. Next slide, please. As you can see from this slide, we rely on contract support in a large variety of areas. The first three are the subjects of today's meetings. We have a number of other contracts that support other activities, such as scientific information management and literature-based evaluations, evaluation of alternative toxicological methods, quality assessment, and so on and so forth. Bioinformatics you heard about in April. We have a number of other contract support mechanisms as well. Next slide, please. Okay, so I've got the translational toxicology pipeline here, and I think you all have seen this many times. I wanted to highlight the fact that the contracts that we're going to be discussing today represent DTT's primary contracted wet lab capabilities, in addition to our intramural labs that I mentioned earlier. And I wanted to highlight the fact that there are a lot of interconnectivities amongst all of our contracts, and relationships such that relationships among our capabilities such that outputs of one become inputs of another. And that sort of is consistent with the idea of this translational toxicology pipeline. And I've starred a number of areas, including bioactivity screening, simple and complex in vitro systems, and animal studies. And those are areas that the contracts we're going to discuss today focus on in terms of our pipeline. Next slide, please. Okay, the next few slides are a build that talks specifically about interconnectivities amongst the chemistry, toxicology, and pathology support contracts. So under chemistry, DTT does activities such as procurement of test articles and characterization of these test articles, development of dose formulation analysis methods, and biosample analysis method development and analysis for internal concentrations. Next slide, please. So under the toxicology support contracts, we do what most people would think of as a toxicology study, spanning from receipt of the test article and some limited chemistry activities through study protocol development, in life and post-life, post-necropsy endpoints, and generation of a lab report. Next slide, please. So you can see here that the chemistry contract provides the test article and the characterized test article, as well as a number of methodologies to the toxicology contract. And in turn, when internal concentration samples are generated, those are sent back to our chemistry contracts for analysis, as well as the test material once that's finished in the toxicology lab. Next slide, please. So our pathology support contracts perform our pathology peer review and finalization of our histopath data. And that's a process that it spans a number of activities, and there's involvement of that from the toxicology lab as well. So next slide, please. So you can see that the data generated from the original read are submitted to the NTP archives, and that those data, and as well as the study pathologist participation in the pathology working group, inform those activities. And then once the histopath data are finalized post peer review, those are returned back to the toxicology lab for inclusion in the lab report. Next slide, please. I want to shift gears a little bit to talk about DTT's centralized management of contracts and budget. So all of our folks that manage contracts are what are called contracting officers representatives, or CORs, and they're authorized by our Office of Acquisitions to perform specific technical and administrative functions under a contract. There's 3 levels of certification, COR certification. The higher levels allow one to manage larger and more complicated contracts. There's requirements in terms of experience, formal training, and a transcript that must be approved by both NIH and HHS. So for example, to serve as a COR level 3, which all of the contracts, or most of the contracts within our division require, one must have at least 2 years of experience on federal projects in addition to the other formal training. Most CORs within our division reside in the Office of Program Operations, and OPO provides centralized oversight of budget and contract acquisition and management to the division, as well as scientific and project management expertise. And you can see that the CORs in our division perform multiple roles depending on the need and in some cases the life cycle of the contract, ranging from acquisition planning to contract and project management, to supporting our project teams and PMTs, as well as serving as resource managers and subject matter experts. So a number of hats that all these folks wear. Next slide, please. Wanted to talk a little bit about, to round this out, on other mechanisms of getting our work done, other than the intramural and contracted areas that we discussed earlier. So there are a couple of mechanisms that we are able to access through our Office of Technology Transfer, including materials transfer agreements, what are called CRADAs, Cooperative Research and Development Agreements, or Research Collaborative Agreements. The government is not able to add funding to these agreements, but they are highly useful for collaborations to make sure things like, materials and publication authorship are notated up front, and there's a good plan for the research. Through our Division of Extramural Research and Training, there are grants and cooperative agreements, the so-called U mechanism. Those require grant funding, which is not part of the DTT allocation. However, once these agreements are awarded, DTT scientists are frequently asked to serve as subject matter experts, collaborating with folks on cooperative agreements. So those are just other ways of accomplishing science that are in addition to the contracted research and intramural. Next slide, please. Okay, I wanted to just get a couple of things down on the slides. Julie's going to go over these in detail next, but just wanted to introduce them here. So as long as our discussions are limited to the review of the general project purposes, scope, goals, and various optional approaches to pursue the overall objectives, the meeting will remain open to the public. If the discussion turns to the development or selection of details of the projects or RFPs, the meeting would be closed. And those if we get into such thing as specific technical approaches, protocols, statements of work, etc. And this is intended to allow for free exchange of the advisory group members' opinions, your opinions, and avoid premature release of details, on the proposed contract projects or RFPs. Next slide, please. And this slide will be part of all the contracts. This is essentially the charge for the BSC for each contract. So asked to review these contracts for overall value, these concepts for overall value and relevance, as well as fulfilling the program goal of protecting public health. And you can see the specific areas under that. Thank you for your attention, and I'll take any questions.
Thank you, Dr. Stout. Are there questions? And thank you for that nice overview of the three contracts that we will be reviewing here. Any questions from the board for Dr. Stout? Hearing none, I will turn that back over to you. So, Dr. Stout, the next presentation then will be on the chemistry support services. Is that correct?
My understanding is that Julie Bardo is going to review some of the procedures from the Office of Acquisitions perspective.
Great.
Thank you, Matt.
Yes, thank you, Matt. Hello, everyone. I'm Julie Bardo, the Research and Development Branch Chief, representing the Office of Acquisition. And as Matt mentioned, I'm just going to go over a few things that he touched on, and a few other things that we want to mention from our perspective from the Office of Acquisition. So NIEHS Division of Translational Toxicology contracts support a variety of activities, including data analysis, literature-based assessments, toxicologic characterization, testing, methods development, and program resources such as chemistry, occupational health and safety, animal production, pathology, quality assurance, and DTT archives and repository. Prior to issuance of a request for proposal, the RFP, also known as the RFP, the review of a research and development contract project concept is required. These contract project concepts may consist of one or more contracts. As today, we're going to be hearing concepts on three different requirements and concepts. Concept reviews for research and development contracts are needed for new contracts, recompetitions with changes in statement of work, and contracts ongoing for 5 or more years since the last concept review. The review of these contract project concepts is conducted by the NTP Board of Scientific Counselors and is open to the public as long as discussions are limited to review of the general project purposes, scopes, goals, and various optional approaches to pursue the overall objectives. The meeting will be closed to the public, however, if the concept discussion turns to the development or selection of details of the projects or RFPs, such as specific technical approaches, protocols, statements of work, data formats, or product specifications. Closing the session is intended to protect the free exchange of the advisory group members' opinions and avoid premature release of details of proposed contract projects or RFPs. The BSC members are asked to review the contract project concepts for overall value and scientific relevance, as well as for fulfilling the program goal of protecting public health. Specific areas should include the significance from a scientific or technical standpoint of the goals of the proposed research or development activity, the availability of the technology and other resources necessary to achieve those goals, the extent to which there are identified practical use for the anticipated results of the activity, and lastly, where the review includes the project approach, the adequacy of the methodology to be utilized in carrying out the activity.
Thank you, Julie. Next, Dr. Eaton, we will proceed with Dr. Saramiya with the chemistry concept. Thank you.
Great, thank you.
Thank you, everyone. So good afternoon or good morning, depending on where you are. As Mylene said, I will be presenting the contract concept for chemistry support for the Division of Translational Toxicology. Next slide, please. I'll start off with a brief history and overview of the requirement. I think I don't have to tell this audience that chemistry support is an essential for our DTT research activities. Because the scope and breadth of this support required exceeds the resources available at NIHS, we have been obtaining these resources for over 40 years through contract mechanism. As you can imagine, the type of support has evolved over the years. This is to align with the division needs. Next slide, please. So this support required falls under 9 broad functional areas. Each of these functional area covers multiple activities. This way, it allows flexibility to support a broader study types or research activities at the division. So I'll take a few minutes to go over the 9 functional areas. The first one is logistics and handling. So this area covers all activities related to procuring, handling, shipping, or even storing test articles. The second functional area is characterization, and this area covers all activities related to identification and purity determination of a test article. The formulation function area, as the name implies, and this area covers all activities related to formulating test articles for either in vivo or in vitro testing. The fourth area I have listed here is biosample analysis, and this area covers all activities related to determining concentration of analytes in biological matrices. The animal studies functional area, and these are related to all activities to determine the ADME and TK properties of test articles. Next slide, please. So the sixth one is a special studies. We call it special studies. So this area covers a variety of a broad range of activities. To highlight the broader range of activities that we have under this area, I have selected 2 examples. The first one is something like determining the partition coefficient of an analyte between 2 matrices. Another broad example is something like screening of multiple botanical lots to select a lot for testing. And the 7th area, functional area, is omics. And this area covers activities related to exposomic, proteomics, or metabolomics. And the in vitro assays functional area, these are activities related to determining ADME properties of test articles in vitro. The last functional area is MHDS activities. And this area covers all activities supporting medium to high throughput toxicity screening activities. For example, procuring large number of chemicals in small quantities, making solutions for testing, as well as something like maintaining a large HDS inventory, chemical inventory. Next slide, please. So I want to highlight here the broader chemistry support required across the DTT translational toxicology pipeline. As you can see, given with the red stars, support is needed to conduct a variety of in vitro testing activities, as well as to conduct a variety of in vivo testing activities, as given with the purple star. In addition, we also need support to conduct both in vitro and in vivo ADME and TK study types. And as you know, that data from this type of studies are typically used either to design toxicology studies or interpret toxicology study data, as well as to translate animal study findings to humans. Next slide. The support that we need typically will be obtained on a per test article basis. And this type of approach allows support to be applied to multiple studies conducted at different research facilities at different times. I'll elaborate that on a little bit. For example, let's say we have selected test article X for testing. And then following that selection, we will complete pre-test chemistry activities, such as procurement, characterization, and developing formulation methods. Then the support can be provided to Lab A during year 1 to conduct a 5-day in vivo study, while the support is also provided to Lab B during the same year to conduct a zebrafish assay. And then subsequently, support can be provided to Lab C during year 3 to conduct a subchronic toxicology study, and then during year 5 to the same lab to conduct a chronic toxicology study. Next slide. This pie chart highlights that the anticipated support can vary per functional area. If you recall, I mentioned 9 functional areas at the beginning. So as you can see, we anticipate that the support to be around 29% for the MHTS functional activities. This is again in alignment with the division, projected division needs. And the support for omics, in vitro assays, and biosample analysis areas, we expect this to be around 15%. And the animal study functional area, again, these are the animal studies investigating ADME and TK behavior of test articles. We anticipate the support to be around 9%. The other areas such as logistics and handling, characterization, formulation, as well as special studies, we anticipate the support to be around 3 to 5%. Next slide, please. So this is a chart to the Board of Scientific Counselors. You saw this slide from Dr. Stout. I just want to highlight very quickly. So the members are asked to review the contract concept for overall value and scientific relevance, as well as for fulfilling NIH's goal of protecting public health. The consideration should be given to the 4 bullets listed here, and we seek the approval from the members to continue this type of activity during using our contract mechanism. And thank you for the time and opportunity, and I'm happy to take any questions you may have.
Thank you, Dr. Wadney and Atha. I have 2 general questions, and then we'll open it to the rest of the board, and then we will hear from Dr. Ryan, who is the lead discussant on this. The 2 general questions I have on the presentation you gave is, I'm assuming that that all of the contract activities for laboratory services have to comply with good laboratory practices. Is that true?
It depends on the type of studies, so sometimes we do conduct exploratory studies, research studies. So in this case, we don't. So our specifications are actually set up in that way. So we have different kind of task types to support a GLP study or an exploratory study. So I would say support both types of studies. Okay, thank you. And the second general question I had was on your bullet item number 7 under omics. It didn't include mention of genomics, yes. DNA sequencing, SNP analysis, that type of thing. Is that part of? Yeah, so we knew that question might come up. We do have a separate contract conducting that. So we try to keep it to all chemical analysis, analytical chemistry related stuff into the chemistry contracts, whereas genomics in a separate. Okay. Does that help? Yeah, thank you. Questions, other questions from any of the board members? Okay, thank you again. Dr. Ryan, you were sort of at the last minute because Dr. Lyon was originally tasked with this and she is not able to attend. Could you please lead the board discussion on this particular contract on the chemistry?
Sure. So thank you for the presentation. And I was wondering if you could help me understand this contract concept. Is it a recompete or a contract that is more than 5 years old? Yeah. Dr. Sorry. Yes, this is a recompete. So I think one of my backgrounds bullets was that we have had this type of contract for over 40 years. So this is a recompete. Okay. And is it fair to say that the newer areas are the omics and the in vitro work? I noticed that represented almost a third of your projected work for the coming year. I'm making sure I can address that question under this forum. Sorry. I can say this much. Nothing has changed from our previous, statement of work. Or again, I don't want to get into too much. I think I would like either Dr. Stout or
Ms. Bardo to kind of jump in if I'm getting into an area that is, So all I can say is that, as I said, these contracts has evolved over the years. Last 40 years. So we are not, so these omics type of stuff we have had in the program, we have done these studies in the program. So it's kind of yeah.
Okay, so then maybe, because I'm also a little tiptoeing around the contract concept here. Could you just help me understand maybe the interface between this omics piece and the bioinformatics concept that we reviewed in April? And then also kind of the interface between the in vitro work that is here, described here, versus what might be in the toxicology concept. So just.
Yeah, certainly. So as I mentioned before, the omics techniques here are very much focused on using the analytical chemistry approaches. So we are focusing on the proteomics, like using mass spectrometry-like techniques, exposomics, and metabolomics. So those are not captured in anywhere else, as far as I could tell. And the second question about the in vitro assays, so the ones I have mentioned here are pretty much focusing on the ADME-TK type of assay, addressing that in vitro, and not necessarily toxicity assays. Does that help?
Yep, yes, it does. Okay. Because there's some, just general terms that we use, but trying to understand what each person means. So I didn't have a lot of other discussion, just those clarifying questions. It seems to me that, having the focus on test article support by individual compound is very efficient. Clearly, the test article characterization and the bioanalytical work is foundational to the other in vitro and in vivo work that's done in the DTT. It appears to me from this high-level overview that you're leveraging available technology to achieve those goals, that there are practical uses for the anticipated results of the activity. And the adequacy of the methodology seems appropriate to me, but I would ask my peers on the BSc for their perspective.
Thank you.
Thank you, Dr. Ryan. I think, Eric, Dr. Bloom, I think you had a chance to look at this as well. Do you have any thoughts or comments on this?
Yeah, thank you. Well, I had a similar thinking than Anne. I was wondering how it fit together, as we think about ultimately leveraging some computational tools, artificial intelligence, and things like that. How are we thinking about the generation of this data, and how do we fit them in the big context of the data streams? And I don't know if it's part of this contract discussion. That's I'm unclear too. But I think it's important to at the end, to achieve our overall goals here, to think about how we can actually get all this data together in a way that we can leverage in a few years artificial intelligence to do better science later. And I don't know if that's part of the contract or not, and if that can be discussed under this forum.
I can start by saying it is probably beyond the part of this particular contract, but I think maybe Dr. Berich can probably jump in, give us a little bit perspective on the data, or someone else.
Hey, Shramya, sorry, Eric, could you pose that question again?
Yeah, I was thinking about, so we're going to generate a lot of data through this contract, and I'm wondering if there is anything that that we need to think about, such that we can more effectively transfer all this data that may be captured in different platforms in a way that help us fulfill our long-term goals of leveraging all these data sets. For example, I can tell you that in my organization, putting all this data in a consistent format in a data warehouse is a significant challenge that is best addressed by thinking proactively along with our partners and vendors how to do it.
Yep. Now that's a great question, and it's a timely question. You may recognize that we actually have a fair history in transparently sharing a lot of the data that we generate in a database that we call SEBS. It's all very publicly available. As we get into generating novel kinds of data, we're actively developing pipelines that allow us to collect that data efficiently and ultimately get it into a central place. So that's just us doing what we usually do. In addition to that, the government more broadly, and NIH in particular, has articulated expectations that not just we do that, but everybody does that. And so we've been actively working through the process of ensuring that all of the data that we generate is available in ways that folks can extract it, use it, and all the kinds of things that you just reflected. We have the benefit of having had a history of traditionally doing that kind of thing. But again, as we generate novel kinds of data, we've got to sort of invent processes that accommodate that. So thanks for that. That's a great question.
Other comments or questions from any of the board on this? So part of our responsibility as board members is to approve each of these concepts through a vote. And I believe Dr. Brownlow will put up on the screen the question for voting.
I will. Before I do so, Matt, did you want to make a comment? I noticed you came on your camera.
No, thank you. Actually, I think Brian well covered it. I was going to mention some of the data management and data sharing plans as well as our SEBS database, but I think yes, Brian very well covered the handling of data. So thank you.
No problem. Thank you. All right. So Dr. Eaton, if you would like, if you can please let me know who makes the motion and seconds the motion. I'll take notes.
Okay, so I would call for a motion on this contract, and this is specifically on the chemistry support contract. Do I hear a motion?
Second.
I'm sorry, I couldn't capture that. I cannot see the screen at the moment.
Sorry. So Dr. Ryan, can you make a motion?
Yeah, so I, as a lead discussant, I'll make a motion that we vote to approve the chemistry support contract as described today.
Second.
And the second was with Dr. Berube. So we have a motion to approve the chemistry support contract as presented. So what I would ask, I'm not sure what the best way to do this is publicly. Dr. Brownlow, you have any advice here? Do we respond in the chat room or just try to do a verbal roll call?
We can do a verbal roll call if that's okay.
All right, so we will go ahead with that. Dr. Berube?
Yes.
Dr. Bloom?
Yes.
Dr. Felter?
Yes.
Dr. Michaels?
Yes.
Dr. Payne Sturgis?
Yes. Yes.
Dr. Rusi? Dr. Rusi? He may have stepped out. Let's see, Dr. Ryan has voted, and Dr. Singla?
She's away.
She had to step away too, that's right.
To step away, she will be back shortly.
Okay.
I'm sorry, it's Mark Rusi. Yes, I agree.
Okay, thank you, Mark.
Thank you.
Okay, so with Dr. Singla abstaining at the moment by her absence, she will be back. We have a near unanimous approval of this concept.
Perfect. Thank you.
Thank you.
Thank you. And thank you for those presentations. So we have on our schedule right now a break. I would suggest that we keep this brief, perhaps no more than 10 minutes. It's now 20 after the hour, so I would suggest that we reconvene at 2:30 Eastern Standard Time and hear the next and the remaining 2 concept presentations. So with that, we'll be on a break for the 10 minutes. Thank you.